So I did my project on the effects of petroleum on egg fertilization success in the sand dollar Dendrester eccentricus. So oil enters the marine environment either via oil spills or via boat fuel spills. When this occurs, a process known as weathering breaks down oil into its components, such as saturated aromatic hydrocarbons and also water-accommodated fractions. A water-accommodated fraction is simply a substance that is poorly soluble in water that is mixed at low energy. This is done in the lab to mimic how oil splits in water following an oil spill. The reason why we chose to look at Dendrester eccentricus is because few studies have been done on how, during its fertilization stages, like how oil affects it. And the reason why we want to look at this is because hydrocarbons negatively affect marine organisms at levels as low as 0.02 milliliters per liter. There was a previous study done on tropical sand dollars known as Melina keatspirata that found that a level of 0.03 milliliters per liter of a water-accommodated fraction of diesel was able to decrease sperm motility. And for this reason, we wanted to look if there was a similar effect in Dendrester eccentricus. So we collected 60 sand dollars from Brady's Beach in Bamfield, BC, on the west coast of Vancouver Island, within the unceded territory of the Hawaiian Nation. We then split these up into 3 experimental trials with 20 sand dollars each that we spawned. Here's a video of us spawning the sand dollars. Okay. Anyways, can you just go back? Let's go back. So we injected them with 0.2 milliliters per gonopore of 0.5 mole solution of KCl. This basically causes the sand dollars to scrunch up all their muscles, and then they shoot out their eggs from the top surface. Up until this point, you don't know the sex of the sand dollar, but after here you can see you get white sperm and red egg. And for this reason, among our experimental trials, we had different initial egg and sperm concentrations. And this mimics what I was talking about earlier with broadcast spawning, where they shoot their eggs up into the water column and they come into contact with these substances. So in order to set up our experiment, we created 4 stock solutions, 2 where the gametes were exposed to diesel and 2 where they were not. We then combined these onto a hemocytometer using a pipette and counted the fertilized cell density. This was meant to see, we did this so that we could see whether or not egg versus sperm exposed to diesel had a negative effect on fertilization to try to separate out those 2 effects. Here's an example of a Dendrester eccentricus egg that's been properly fertilized. You can tell by that little red layer you see around the edge, the dots, that's known as the halo layer. And here are 2 examples of eggs we wouldn't have counted because they're unfertilized because there is abnormalities in the halo layer. We then compiled all of our data prior to the creation of this graph to see if there was an effective treatment. We did not find an effective treatment on fertilized cell density, so we then went and we separated out into our experimental trials. This was in part done because when we ran linear models to see whether treatment or time of exposure or experimental trial affected fertilized cell density, we found that experimental trial had the greatest effect. And when we graph this out, we see that experimental trial 2 in particular seems a little bit different than the other trials, and this is unusual if you consider the fact that experimental trials 2 and 3 had very similar initial egg and sperm concentrations. For this reason, we cannot conclude an effect of diesel on fertilization success in Dendrester eccentricus, and we have to assume that it would be unimpacted by an oil spill. Of course, we have to be cautious about this since this is the first study of this nature, and it was done on a low budget at Bamfield Marine Science Center. Future studies could look at higher concentrations of hydrocarbons because maybe the concentration of 0.03 milliliters per liter that we used was not enough to elicit an effect. They could also look at different sand dollar populations, since we are just sampling from one population here, to see if there was differences in how their hydrocarbon tolerance was. Thank you.